Hello, everybody. So before we get started, I tend to go a little fast. If it goes too fast, raise your hand or something. Tell me to stop. And if something I'm saying doesn't make sense or you have questions, please feel free to stop and discuss things. One of the caveats with behavior in general is it's really easy for me to come up here and say you should think this and think that until you actually get your animals in there and you start practicing in whatever area or lab that you have. You'll find that there could be a million of other things that could go on or you'll come up with or ideas that will happen. Let's see. So today what we're going to discuss is what criteria should you look at or consider before you choose your test? And then I'll present some commonly used behavior procedures, and then examples of how to use these assessments. Most of these are familiar to me. There's some that are not. I do have other behavior experience, but all these are going to be very simple locomotor sort of based behaviors. And so why do you need to do behavior? You have various reasons, such as if you have a loss or gain function of studies, what therapeutic potentials are you looking at, whether it's drugs or knockout animals or a particular gene you're expressing or taking out. And then when you do these alterations, do they have side effects? Are there motor or behavioral changes? Or even, like if you are doing a drug, is the dosage the right dose that you're using? Are you seeing an effect because it's too much? Or are you not seeing an effect because it's not enough? Or it's just not working? And then secondly, especially when you are doing genetic manipulations, you need to know, does this knockout have any, or knock in, does it have any behavior outcomes that you weren't or were expecting? And then, of course, in the end is always to make your reviewers happy because you want to publish your work and you don't want them to come back to say, well, is this really because of this or is it just because they didn't want to move, you know? So firstly, why do we do all these research here? I'm in more of the sort of translational health effects for humans. So, you look at assays to assess drugs of abuse, memory. I'm currently in a traumatic brain injury lab, as well as anxiety or mental disorders. And as well as things such as spinal cord injury and other neurological disorders that affects locomotor movement. So how do we get to this point where we can help humans do this? And that's all, not all, but mostly through behavior animal research projects. And so here, of course, your techniques are going to be determined by what animal model you're interested in using. And so you can see here from the C. elegans. Can you guys see that? Huh? No? All right. It's working. Okay. So you can see here, you can go very simple, such as C. elegans. They have around 20,000 different genes, rodents, and then all the way down to humans. And you can go from simple to very complex models. And this will have a big say on what kind of assays you're going to do. So you're going to start off with, examining your particular animal model of interest. And then when you look at these as well, you got, if you're, say, looking at actions of drugs, does that animal model even have the receptor that your drug works at? Because I think it's like pigeons or something. If you do morphine studies, pigeons are mostly more kappa receptors versus the mu opioid receptor, which morphine is active on. So you just, you have to keep in mind these other aspects. But when you first start your behavioral study, there's a couple of criteria that you need to consider, and they're subdivided into about 4 groups. You got validity, reliability, sensitivity, and utility. In academia, what you're probably going to hear, first of all, is how much is this test going to cost us? do we have the funds to do this test, and or space? And so that's, you know, one of the things to think about is it cost effective? Is it easy to run? Do you have to train yourself, the animals? How much time do you want to keep the animals around? How much training do you need? Is this something that you are the only person that can do this because of how much training is required? So you'll end up doing this test for everybody in the lab, or, you know, things like that. But further, you need to look at validity. And there's 3 types of validity that you should consider. And it's going to be difficult to hit all 3 on your particular assay. But it doesn't mean that it can't have at least one of these. And then, so first is going to be your construct validity. And then, so what this basically means is the output you're seeing, does it follow the same neurobiological processes that underlie the disease? For example, like Alzheimer's. Are you seeing your response in your assay because it's due to plaques in the brain, or is it due to the fact that you lesioned something? And then second is face validity. Does it seem similar to human movement? For example, drugs of abuse. You have self-administration, which would have more face validity, because self-administration means the animal is voluntarily taking the drug, versus where you do like condition-place preference, which is predictive validity, which you are giving the drug to them and they're still giving the same response that you expect them to have, but you're forcing them to take the drug. Does that make sense? Yes. And then next you want to do reliability. Is this a test that if I do the test and then somebody else in the lab does the test and is blinded, are you going to get the same results? Or are you going to look at, are you going to see completely different results? And is it sensitive enough that if you move your apparatus from here to here, you're going to get 2 different responses? These are, and then, so you have to think about those things when you're looking at your test. You know, if you're in a very busy area, a lot of noise, a lot of traffic, you don't particularly want a test that will cause them to, if they need to move around and then they freeze because they hear a thump of a door, or the elevator's going off, or people talking on their cell phones. So these are other things that you need to think about. And then also sensitivity. I did a lot of condition-place preference, and so it wasn't sensitive because it was very variable. So you, depending on the breed or the strain of mice or rat you used, you could have, you might not have enough animals. So like, for example, if you do cocaine, you might only need an N of 4 to get your results. You do nicotine, you might need an N of 10 to get, to see significance or preference for that, just because of the variability. And then, of course, thirdly is utility. You know, is it cost effective? Can you, so during my postdoctoral time, I've learned to make apparatuses versus buying a $10,000 apparatus. You've kind of learned to put wood together and put cameras on there and be an eBay shopper. So it's, you know, it's very, you know, one of the questions is going to come, can you implement it easily? Does it require a lot of training? Do you have the training to do it? Do you need to go somewhere to get the training done and then come back? And then these are just things that are inherent in any kind of assay that you would be starting up. So for the first example model, I have C. elegans, and this is courtesy of Dr. Jill Bettinger. She gave me the videos to do this. So C. elegans are really cool to look at mobility and locomotor movements, and also it's a very quick assay. I think it's 3 days from breeding to the time you can use the animal. So it's very quick, and and they're very easy. You just put whatever chemical or drug or you're testing right on the plate, and they soak it up. And you can see here with these wild type worms, see how they make these deep bends. I'm sorry, the mouse is getting to me. You see how they make these nice deep bends as they move. So you can alter that genetically to take out that physical movement. And so what can happen is, so say you have these same animals, and now they're on ethanol. So ethanol's pretty much made them drunk. They can't make those deep bends. They can hardly make forward progression. They're not moving anywhere. Oh, that one, he's doing well. So it's kind of interesting. So why do you see that? And so you can knock out a particular gene and see if you can lose that phenotype in that animal, and then you can say, well, maybe this gene is what causes, development to tolerance of alcoholics, say, or and then so can you alter that gene? Can you make a drug that targets that gene? And that's just from this very simple locomotor behavior test. And then, so rodent models are much more up my alley. So we'll be looking through quite a few different ones. And like I said, if you guys have any questions, stop me and ask. Or if this is not quite what you're looking for information-wise, I can try to help you with that. So spontaneous activity is a very easy test, very simple thing to do. You can, and it gives you a lot of data. It gives you a lot of different things. So when you put the animal here in this box, depending on what kind of box you have, it can have photo beam breaks, which can tell you how many times it goes across the box and breaks the beams. It can tell you exploration habits. So, for example, when a rodent is put in the box, they're going to immediately want to go along the edge of the box because they're prey animals, so they want to sort of stay covered. And that can also be a measure of an anxiety-like phenotype. So from this, you not only get how fast they're moving, where they're moving to, but you can also sort of get a feel of how anxiolytic your strain of animal is. And then you can see that once you say you give them something, well, they're suddenly less active. Or they can become more active. Or you can see that they spend more time along the edge of the box versus exploring to the center of the apparatus. And this is especially helpful if you're doing this because any other behavior test that involves movement, is it because the results you're seeing is because this animal is suddenly hyperactive or hypoactive, or they just are have increased anxiety-like phenotype? And so here's a nice, I should have loaded these before, sorry. Here's a nice, so you can see here, the animal on the right is a wild-type mouse. He's just sort of hanging out, not doing much. Now here's a knockout mouse, and you can see suddenly how much more he moves around. And this could be a particular confound in your test, especially if you're looking at an animal, I don't know, that goes from one location to another, and you want to see how fast it's going compared to a wild-type mouse. Well, is it going that fast? I don't want that. I don't know what that is. Oh. Let's see if I can get it to come back. There we go. So this is very important, especially if you're doing something that is dependent on locomotor movement. Because if you have an animal that then doesn't move any time, and you're getting this nice score, oh, the drug, prevents them from doing this. Well, is it because they just don't move, or because of the drug? So this is a very important first step. Next particular apparatus is beam walking. This is used in a lot of traumatic brain injury labs, and there's two different kinds. The first one, you're kind of just seeing, okay, well, right here there's going to be this really bright light and this like 100 decibel noise that's really loud. I had to get like a sound meter to figure out, like, it's crazy noisy. and they're supposed to run away from it and go straight into the box. So what you want to do is time how long it takes them to go across to the box. And of course, it's really easy to do. There's a pre-training day, and then you can do your injury, and then depending on how many days out you want to do the study. Now, something that we ran into, and then in this case, you want to look at paw placements when they have these pegs sticking out. So you want to see is their coordination. How easily are they able to go around these pegs or over the pegs and stay on the beam? And it'll be the same thing. They'll have this really bright light and a loud noise, and they have to run away from it. So I just recently had experience with this, and we found out that in our case, our rats were looking at us. So they followed us on the beam. They didn't like go to the box. They were looking at us and reaching out to us to take them off the beam. So that was a really big confound. So we basically had to not be in the room with them and hope that they stayed on the beam. But so that was one of the, and then another ones we had is they went towards the noise and light just because it seemed safer for them because they can go hit this table versus going across this really high beam. But for pro, it's very easy test to do. Doesn't require a lot of manpower to do it. Very quick test, 5 minutes. You've done 3 trials, and it's done.
Is it really just the amount of time that it takes them to walk the beam, or really what are you actually looking for?
So you're doing a couple things. If you look at a TBI rat, so a non-TBI rodent rat will cross the beam in like 3 seconds. When you have a TBI animal, it will take him 15, 30 minutes, a minute to cross. And then, so from there, you can manipulate it, right? So you can say, well, I gave him a drug. Does it improve that time? I did a knockout. Does that manipulation do a difference? So that's what you're trying to look for is recovery from motor deficits that are involved with whichever you do, if you do stroke or TBI or cardiac or anything that involves this motor component.
So I've never done this, but my bosses are interested in getting more locomotor tasks. And in your experience, how profound does your stimulus have to be to get a deficit in this? So like TBI, and you've got minutes difference, but I wouldn't expect with our protein that we would necessarily have profound effects. Would you recommend it, or do you think that this is better for?
Does it show any sort of?
Yeah, you'll get like we can see Rotorod or just total activity locomotor differences. But
It might be. You might not want to do like directly the straight beam walk.
yeah.
But if you do this one where you kind of make an obstacle for them so they have to balance and know where to place their feet. So for like the TBI animals, for this study, it affected a lot of their back leg movement. So what they really did was they took their front legs and sort of pulled themselves across the beam. There's a compensation change. And so at this point, our confound came, do we still count that as them actually getting through the paradigm? Because they didn't use like all 4 feet or, so that might be more of what you would need to see. We currently set up a camera, so now, yes, we can watch them on the video. We also time them during the test, because it's very simple. It's very straightforward. It's not a lot of watching, but we do both, yes. So you can do either or. We've not set up, you could maybe set up a program where they, like an Maze or something like that, so it can automatically calculate the time they take to go across the beam. I just don't know how it would work with like foot placement. That might be something you would have to watch physically. To answer your question?
Yeah, I guess I find it easier to score a little bit easier tests because sometimes you have to move angles.
Yeah, so we're kind of lucky because of how our room is set up, that it stays in the same place and the camera can take a wide range. but we also do it, for this case, we do this also live. We do locomotor and other tests that we do video and look at later, and you're like kind of kicking yourself because the camera moved a little bit or something like that.
How do you register these misplacement stuff? Like you see how many times he slip off?
Yeah, so what you can do is, so what they kind of do when you first see them do the trial after TBI is that as they're walking, you'll see that they'll literally like do a back split. Their feet will just totally come off the, and that would be considered a fault, a foot fault.
So whenever you see that, you just tick mark?
Yeah, you just tick mark that they had a foot fault. And in our case, we keep them around for a couple of days after the TBI, so you'll notice that those foot faults will decrease, over time.
So how much time it takes for them to train when they're normal, and once you induce the disorder, what happens after that?
So when we train them, we do 1 day, 1 day of 3 until they get under 5 seconds.
Okay.
And some of them, this is where your baselines are important, because some of them will take 3 trials to do that, and some of them will take 10 trials to do that. Why they do that, all we can tell is that some just learn. I think it's somewhat of a learning aspect too, because they have to know they have to get from here to here.
Right. When we do our experiments on a normal rat, we don't even have to train them; they just do it.
Mm-hmm.
We have a very similar arrangement. But when they got the motor disorder, they don't want to do it, even with big noise or something. So we were just wondering how. So even though if we train them a lot when they're normal, they won't do it once the disorder is there.
So that maybe depends on the severity of your disorder. We noticed that, so we have depth of how traumatic the brain injury that we do is, and we noticed at like 2.5 millimeters in that they weren't doing this test.
Yeah.
But the first day they didn't do it, but the second day, or the third day, or the fourth day. Yes, so behavior takes a lot of patience and energy and time. They're very simple tests in theory, and then once you get in there, that was the caveat. What depends in your lab, what works, and does it work with your particular assay? Could it be that your disorder is too much for this particular assay? Could there be another one that you could do?
Do you have these guys on a reverse light cycle, and do you do this in their dark phase?
No, so that is also something you should consider. We do this during their dark phase, I guess, because we don't have them on reverse light cycle. Would they do something different? Maybe. I don't know. We haven't tried it. We keep all our animals at the same level. But if it's something that you're interested in and you want to see those differences, then that's something you should keep as well, the same for you, however you would prefer to do it. If you have animals that drink, you do alcohol studies, so they drink more at night. So to see them drinking during the day, you probably put them on a reversal dark light cycle, so they're dark during the day and light at night. That could be something that you would have to do. The only thing is, like, we run this in the dark except for that bright light.
Yeah.
So they are in the dark, except for the bright offensive light that they're supposed to run away from.
Yeah, that apparently can cause a phase shift, though, can't it? Cause of jet lag. Just even having that flash, is that right?
Not for the amount of time.
No, yeah.
How long is the task? 5 minutes?
Yeah, because you do like 3 trials. They rest in the box for like 30 seconds. It takes you about 5 minutes to run the trial. I mean, you could have this jet lag kind of phenomena, but if you make it all the same, you will compensate. Don't run some of your animals at night and some of your animals in the morning and some of your animals middle of the day. Or if you do that, you need to make sure it's sort of randomized, so not the same animal gets run in the morning every day, or same animal gets run in the afternoon, but you sort of randomize it. so you get an idea overall if you're seeing some sort of effect like that. And then, so this is an example of data that uses looking at beam walk. So there was this idea that antipsychotic drugs to manage traumatic brain injury and then they show that these administration of these hindered recovery. And so they wanted to see if giving these antidepressants had an effect on motor recovery. So they had an effect on emotional recovery, but they wanted to see if there was an effect on motor recovery. And what they found basically was that, if you look here, here's the pre-trial. All the animals took like 5 seconds to cross the beam walk. And that's something else too. You need to consider your criteria for excluding an animal that hasn't had anything happen. Are you looking at 5 trials? If it doesn't get under 5 seconds, then this animal is not something that is not one that we want to use. These are things you need to consider beforehand. And it's always good to do your pre-baselines for this. And then you can see here is that, this is your sham animal all the way through. The sham animal means that it basically didn't have the TBI injury, but it went through the surgery. It went through the full surgery for it. And then as they give the different drugs, compared to your TBI vehicle, which is the darkened circle, you can see there was no effect of the drugs improving the motor coordination of these animals on beam walk. And so you can see that, in this case, the drug had no effect. And you wouldn't have really known that unless you, kept your control. You had your sham animal with your control and then your TBI and vehicle.
So the mice, they have remember. I mean, after you treat the animal, and they repeat each day, right?
Yes.
So they remember something they can do better, I mean?
They should, yes. So the idea is that you're supposed to give them that memory before you do the TBI. So they should have the idea that okay, I'm here and I need to go over here. And you should do that prior to any kind of treatment. And then when you then do the TBI, now your question is, are you impairing learning or are you just impairing a motor kind of thing? That is something that is, it could be yes, it could be no, could be, but as long as you have your controls, then you should be okay. And then another analysis you could do is Gait Analysis, which looks at motor function and coordination. You have this really cool thing called a catwalk, and it's sort of basically like this treadmill, and it can, it gives you paint their feet, and it can take, it'll take computer measurements of their stride and the length of their stride and how do they walk and things like that. Or once again, like we go back to that chart, you got to think about that. That's a pretty expensive kind of apparatus to do. Or you can just take like bench paper and paint their toes with nail polish and have them walk in a, have a little corridor for them, and you just have them walk straight down. And you can also measure these different things. And this doesn't require, as you said, per se more learning. This is just very basic motor, and this is in assays such as TBI or stroke where the motor coordination is hindered, or even your HIV studies, right? Is that this one might be more feasible for you versus the other beam walk. And then there's Rotorod, which is really a very simple test. It also requires some learning. So you just basically turn on this motor and it rotates this wheel, and then you put the rat or mouse on there, and you time how long it takes for them to fall off, or just they'll hold on, they'll grab on and just spin with the wheel. And you can do different things. You can do fixed or accelerated speeds. So you can do like a fixed speed where they just stay at the same speed. You can do accelerated speeds where you increase the speed in increments, and you see how long they can hold on at a certain speed. Of course, the advantage of this is that it's really simple to use, doesn't require a lot of resources. It is a pretty sensitive test. But there's some confounds to this, like they'll cling to the beam, they'll refuse to test. The weight can have an issue. If they're quite heavy, they might not be able to pull themselves around the core. Or in cases of accelerated speeds, they might be tired. Well, why are they not holding on at the higher speeds? Is it because they're tired from the previous speeds? So these are things that you have to. We had one when we just did this. After TBI, he learned that if he waits for the wheel to go backwards to a certain spot, he'll just hold on. He'll let go of his back legs, hold on by his front, and then let go and drop into the cage underneath him. So it's really interesting because this is one I talked about, and then I was like, oh, this is easy enough, and then I actually did it. And so we had to find out that we couldn't do 3 days of training pre-training, because by the third day, they even refused to, they would twist their bodies and refuse to go on inside the thing. So these are, you have to learn your species of animal and strain, and is it willing to do that test that you keep putting him on, or is it going to be like, no, I know that if I fall off, you're going to tell me I can stop. So here you can watch this pretty cool video. And that's something too. So they have a mat around here. Some have bars, so they can like just hold on to the bars and decide to spin around a few times. And then age can have an factor in this as well. If you have older animals or age-induced animals. So that's my favorite guy right there. And so you have to decide, do you end your test when that mouse or rat does that, or do you wait till he does it 2 times around, or, do you just put him back up on there if they fall off? So in some cases, like our rats, we found out that we had to put him back on there so he knows that just because he falls off, he's not done.
So the only measure that we get from this is the latency.
Yes, latency to fall off. And so this, this looks at motor coordination. There's also some learning involved in it. You'd also want to do a pre-baseline to see if your animals are able to do this. Some don't even just hold on; they just fall right off.
So in your experience, how much training do you want for Rotorod?
We did 1 day.
1 day.
3 trials, and that was it. And then we did accelerated speed, and so the total time we allowed them to be on there was for like 1 minute and 45 seconds. Was like the end of the cycle. We didn't keep them on there longer than that. What we noticed is that prior to injury, they all did that. There was no real issue with them doing that. Now, some of them were kind of like, well, I'm not going to do this. I'll just walk right off and fall. And that's what they did. So you can't obviously use that animal. Either he's too smart for us, or He was, yeah. Some of them are quite smart. We had one that would rear up on the low speeds and then turn around and just kind of walk down the rotor rod. So that's also one that you might want to think about is can you adjust the spears that hold them in? So we had to kind of make them sort of tight so they couldn't just kind of turn around in the apparatus. And it was very much like, oh, this is easy enough, right? We can just stick them on here and you put them on there and we can see if the TBI had an effect or the drug. In our case, we were using a drug. Did it have an effect on the amount of time that they spent on the Rotorod? Well, what we found out is that, one, you can't train them for 3 days prior to that because then they'll refuse to do it. Or if you do it every day during the treatment of the drug, that by the 4th day they're really bad because they just don't want to stay on the Rotorod. But you know they're actually improving, but you can't show that because they have their own ideas. And then, so those were sort of like basic interfering with motor coordination kind of tests. We can also do in rodents, you can do multiple anxiety-like tests. Two of the ones that I'm very familiar with are the light-dark box and elevated plus maze. And both of these kind of give the animal a conflict because they're very curious animals. So they want to explore and see what kind of environment they're in. But they also want to stay in these dark, closed areas because they are prey animals. So if they stay in the closed, dark, they're not going to get eaten by something. So what you want, and then in the case of the elevated plus maze, you add the height factor to it. So you've now put them in, they're scared, they're on this high, little short beam, and then you're also involving this need for them to explore. And these tests are considered the anxiety-like tests because they're sensitive to treatments with benzodiazepines. So if you administer an animal with benzodiazepine, you'll find that they will go out more in the light area. They'll go out more into the open arm. And this is good because it's you can do a quick screening of your test drug if you think, well, is this going to have an anti-anxiety-like effect on the animal, or is it going to remove, say you have an animal going through a drug withdrawal, and you'll know an animal going through drug withdrawal will not explore their area or anything. So you're going to give them this a therapeutic agent. Does it block that part of the withdrawal? And this also has this inherent also locomotor behavior component. So if you give them a drug and they just stay in the dark place, is that because that's where they want to stay, or is it because they're not moving very well or the drug is hindering their activity? And you can do that by you'll see you can count by like the number of crosses they make, because even if they're staying in the dark, they'll go back and forth between the two arms, or they'll be active inside this box. And so you can look at those to look at if it is indeed helping or not helping, or is it because of motor effects. The disadvantage of this test is that it is influenced by various things. So the species that you're using, housing conditions, daytime of testing, how much light you have on the light side. You can make it so aversive that they won't go in the light, or you can make it not aversive enough that they just decide to hang out there the whole time. And then how you score it. You have to make sure that you're consistent in what you're scoring within the lab, and that you have a certain protocol that says, okay, well, we're going to count. Example, in elevated plus maze, we have an exploration behavior called head dipping, where they actually go under, they lean over and look underneath the beam, which is considered an anti-anxiety like behavior because they're actually exploring and coming out and looking around. But another lab may not count that. You guys have to be consistent in what you decide that you want to do. So the next one we look at is you can also do various memory tests, and you have this in case is the Morse water maze, which is spatial memory. And it's not an easy test to do. So that's one of its cons. It takes a lot of time. It's space intensive, but you can do multiple things with it once you've trained the animal. So you can look here, once you train session 1, the animal has to find the platform, which is hidden under like this latex white paint. And they have to find it. And then by session 4, you can see that they found it. Well, what if you did something to the animal? For example, say, I have an example. We had Alzheimer's mice, okay? I didn't have Alzheimer's. Another lab had Alzheimer's mice. And they're training them, and they find early on that they're able to find the platform. But then they slowly realize that they can't find the platform as they continue on. As the animals get older, they stop being able to find the platform. And what they actually found out was that it wasn't really the Alzheimer's phenotype that was causing that, it was their eye vision. Because they have to have spatial cues around the room to be able to see and remember where the platform is. And so what it found out is that they had degenerative eye disease. And so they couldn't see those cues around there. So that's one of the things that you have to take into consideration. If something involves these visual cues, does your animal or strain have any issues with eye, with seeing or vision or things like that? And then, too, it could also be a memory thing. So, for example, for TBI, once you do TBI, you'll probably see something more like this versus this. And then you would want to see, does a drug help that memory improvement? And then we do another one called object recognition, and this involves more working memory. So when you train them, you have them in a box, and they have 2 objects. And this is 3 days. We do all this in 1 day. We put them in here, then we take them out for 5 minutes, and then we replace one of the objects with a new object. And then we investigate the time they spent, or we analyze the time they spent investigating the new object. And we also do this with, we use this more as our attention disorder paradigm. And because I also work with secondhand tobacco smoke effects in neurodevelopment. And so we use this one more so to assess if the animals can understand that they have a new object within their environment. And of course, this has, it's really easy to do. You can repeat trials, you can change up objects, and it's inexpensive. You just need some toys. We have cats for the mice, for the rats to investigate. Cons is operator error. If I do it versus somebody else who does it, where do you place the animal? Is the animal placed in the same position in the box? Do they face toward the object, away from the object, in the middle of the object? Or things like that you have to make sure that you are making standard in your lab. And then we have some feline models here, which are mostly used in spinal injury. And they're used because of their ability to recover from hind limb injury, to recover hind limb locomotion following spinal cord injury. And you can see here that these animals who've had spinal cord injury can do this peg walk across this, and they're able to assess various paw placements, limb trajectories, and this is also very cross-species. So you can do this in rodents, mice, primates, non-human primates, and then this will all translate to human therapies. And then finally, this is like hints to avoid confounds. You need to think about your environment from the animal perspective. Is your animal nocturnal? Does it have a large olfactory bulb? Is it going to be more sensitive to smells? Do you have blood or signs of natural predators? Noise level in your building, like do you do a study on a weekend versus a weekday? construction, food smells, perfumes. I try when I'm working with animals that I smell the same the whole time through with the animals. I use like scent-free detergents, deodorants, no perfumes, no perfume lotions. I have animals at home. I have dogs, which are predators, and cats. So I make sure like all my clothes, the animals don't sit on them. They're packed away in the drawers, that kind of thing. You need to control, you need to make sure, did you see, for whatever reason, there was no food in the cage. How long did the animals go without food? You might want to make sure that when you test them, did something, are they off? Is it because they didn't have food, or their water bottles leaked? Were they in a wet cage all weekend? Or secondly, do you run your animals in the same order, or do you randomize your animals? And then, of course, expect baselines in your lab. One of the biggest things we notice is we had condition place preference in one building, was doing fine. We moved to a different building. None of our results were the same. They weren't as robust or as high in the new building compared to the older building we were in. So it's just, you have to know what your baselines are for that particular environment. And then, so I made an example of what happened to me as a graduate student. So basically what I did this condition place preference paradigm, and it's locomotor based, so they have to be able to move from one box to another. And we found that, I did nicotine, so they were paired, say, nicotine injections were paired with this white box, and then the vehicle was paired with this black box. And I found that, nicotine, they always went to their paired side of the box. But when I gave them this enzyme inhibitor, they didn't prefer nicotine anymore. They were sort of baseline in both boxes. So they didn't have this nicotine preference anymore. So one of our reviewers came back and said, so this was my results. We did 2 different doses. We found at these 2 doses, or at this last dose, they significantly decreased nicotine preference. And we saw that, both of these doses by themselves didn't really induce a preference. But the question came back is, well, they're not inducing preference or blocking preference because the inhibitor is blocking movement. So, of course, we had to do a locomotor activity test on these drug doses with and without nicotine to prove to them that the results that we're seeing wasn't due to the fact that they are not moving anymore. And that's it. And then I have this video here if you guys want to watch it. It's about the 3rd reviewer. Hey. Thank you. You guys have any questions?
If you're doing some activity assessment by a video tracking system or something like that, do you know of any good video tracking software on a budget?
Okay, you got the keyword is on a budget. Our lab uses Maze software. It's not very cheap. It's like $5,000 or something like that, just for the software. But that being said, it gives you 5 licenses or something. So you could share it between labs maybe, you and another lab could go in. That's how we did it. We shared it. Whoever gave the more percentage got 3, and the other one got 2. That's kind of how we did it. I have yet to see one that's cheaper. Now, we save some money by putting in our own, we use a video camera security system kind of purchase with a DVR recorder system, and we were able to hook up the cameras. But we don't have the Maze software hooked up to those cameras. So we have to take the videos and then put them onto a separate computer with the Maze on it. So that's how we saved money, because that whole system is about $10,000 or something.
Yeah. That's exactly what I had in mind, is getting some cheap little cameras.
Yeah, and we
Worry about all that
got the
stuff.
infrared because we do a lot of ours in the dark. So the infrared helps us to see the animals. And then the DVR is really nice because then you can decide what file format you want to put the videos in and then put them in Animaze.
I feel like a lot of these kind of tests are kind of things that, you want a good way to hack a sort of preliminary experiment together so that you can show a proof of concept, and then, see about budgeting for more professional looking equipment or things like that.
Yeah.
So do you have any advice for like the kind of hack together an experiment sort of perspective?
Hack an experiment together perspective. So like say you're trying to get a pilot data for a grant,
Right.
basically. So this is kind of, that depends on like what you're trying to show and how sensitive it is to show that. For example, our TBI animals, we noticed that because we used an outbred strain, that an N of 1 wasn't going to be enough for our pilot data because they were very varied in their response. Some of our animals who had TBI acted like they never had TBI, and some of our animals acted like they had very severe TBI. After crossing out other issues that it could have been, it's just come down to the fact that it's like if you or I or you had TBI, we would all respond differently to it. That's basically what we saw. So you might look at, so now we're going to go like with an N of 3 for each assay that we're doing. Does that make sense? Is that the question you're asking? No.
Can I say this one thing too? Is that of all the tasks you showed, the most versatile box that you will get a lot of usage out of is the open field.
Yeah.
Because you can run that for 30 minutes and use that as a locomotor assay. You can take the first 5 minutes of that and utilize the center time or center entries as an anxiety task. And you can also do the object rec or object placement tasks in that same box. So it's a lot of bang for your buck out of that. And I suppose you could retrofit it for a light dark.
Yeah, we did too. So we made inserts that made half the box dark and the other half was light. So we added extra lighting that goes into that. And then we have like one big box that we put into 4 quadrants. So we can run 4 animals at a time because we have the video camera running. And so that's just things definitely. Like you can also do another test that I didn't put on here that we do is also social interaction, which is a very easy test. You put an animal in a slotted cage kind of thing in the center, and then you see how much time they spend with the animal.
So I'm thinking, like, what we're doing is, motor assessment. So I'm trying to figure out, like, where is, like, the most immediate best bang for your buck? Like, what, the open field study, or, like, is this elevated beam, or,
I kind of like the idea that you gave for gait analysis instead of using that expensive instrument, paint their paws and take their and make them walk on a white paper or something, and then study that. That's a much cheaper option. We could go for it. Do you have any suggestions for to objectively measure the posture abnormalities?
So if they slip off
No, for example, the rats that we lesion, sometimes they show some tripod postures, really crooked spine. So is there any way to measure
that objectively?
Yes.
The only thing I could recommend is that you videotape these postures that you want to record, and you kind of make a standard in the lab that if you see it this way, this is how you're going to count it. If you see it
or use
this
these
way.
markers that you showed in that cat experiment, and use the image analysis to measure the angles.
Right. So you just want to make sure that you have standardized what you want to look for, and that one person doesn't say, well, he was kind of this hunched position, but not really. So do we count him? You know, so it goes back to like how angled do you want that hunched position to be? What is your cutoff to count it as a symptom or endpoint? So that's part of these behaviors is kind of, in the end, you see, you know, the pros and cons and what people do, but in the end, you're gonna have to see what works best for your particular study.
Yeah.
Which is not much
and then also how to satisfy the previewers that we are doing
right. So one of the
more objectively.
One of the best things I've done is look at other papers and what was their, how did they catalog? How did they say, okay, this is that, this is that? And then if it comes down to it, email the author, because they always have more methods than what they put in the paper. So it's always great to email the authors. And you'll find a lot of them are pretty responsive. You'll find the few that don't respond back, but in most cases you'll find that at least the first author or second author will respond pretty quickly, if not the senior author on the paper.
I have a question. What kind of preliminary information do you have to have before you can give a new compound to an animal? Like, do you have to have in vitro data?
That's a big. That's like a pharmacology question, which is fine. There's a lot of things you need to take into consideration when you're giving a compound. That, you know, can you find data on it? Has somebody published anything on it? That's going to be always your first step. If you found that it's only done in vitro, then you're going to kind of be in a new field. You're going to have to see, can I extrapolate the doses they used in vitro? Can I make them into animal doses? You know, which you can do. And then you're going to have to figure out what kind of injection method do I need, and is that based on the vehicle that it has to be dissolved in. So if they are using, for example, we just use something called methylcellulose, and that is oral gavage administration. So this drug that we found out, if dissolved in this, can only be given oral gavage. So now that's your next step, you know, is can I train the animal? Can I give this oral gavage? Can I be trained to do oral gavage? And then your doses, that's where you kind of have to say, do I have the funds? Do I have the money? Do I have the time to test how many doses do I need to see? Ideally, you want to do at least 3-4 doses would be wonderful. Sometimes maybe you only get one chance and you do 2 doses, you know. So that's kind of where you're at on a drug that hasn't been tested in vivo. Now, you can be lucky if, say, you have it in a particular model. So, for example, this drug that we're working on in our lab right now, it's been done in spinal cord injury, but not TBI injury. But you have a whole animal experiment. You have doses that they gave. You can maybe go from those doses and translate it to your study. So does that answer your question?
Well, I'm thinking more, I mean, for some things, yeah, but I'm thinking more of what if you made the compound yourself and it's completely new.
Oh, okay, right. So I guess you would have to look at, so in vitro data would be nice. Do you know what receptor it binds to? What is the potency of that receptor? Which you would probably know what you think it would bind to, right?
Yeah.
Based on what you made and its, you know, KD values and things like that. So you should be able to extrapolate from that what dose you think you should give them. This drug that we're doing too was somebody made it in their lab, and then they went and did their animal studies based off of that. And then now we're taking that drug and using those slightly different doses based on what we are compounding for. But, you know, one of the biggest things you want to do is maybe do a dose and then see if it gets into the brain, if that's where your target of interest is, you know, do some mass spec on it, see if the drug is in the brain or in the blood or, you know, what is its half-life.
I think there are vets here at VCU too who could advise you on what to do.
Yeah, that's true. Yeah, you could talk to Bobby Collins is a great person to talk to, or the vet techs. Stacy and Cindy. They're really great people to talk to about that as well.
Great. Well, thanks everybody for coming. There's more snacks, so help yourself. And thanks to Dr. Muldoon for her presentation today.